Thank you for inviting me to give this talk, and I apologize for not being present in person at your meeting. In this presentation, I will share my thoughts on the place of plasma products in the development of blood systems globally, with an emphasis on countries where blood systems are still under development. I wish to point out that the situation of blood components as the form of transfusion therapy pr- practiced in most Western countries is not universal and is dependent on the economic status of each country. In this slide, data for the World Health Organization demonstrates that while most blood donations from upper and high-income countries are processed to components, this situation is different in low and middle-income countries, where a much lower proportion of donations is processed into such components. I want to point out that the historical establishment of blood component production has not been part of some universal principle, but has occurred in different countries for different reasons. In the United States, for example, the replacement of plasma by red cell preservative solutions occurred because of the need to minimize red cell expiry by extending red cell shelf life. This was needed in the 1970s because of the great shortage of blood which occurred when paid plasma donation was banned as a result of the work of Richard Titmus. The collection of plasma in the United States has not been from whole blood collection, but from the apheresis of paid plasma donors. In the United Kingdom, Australia, and many other public health blood systems, plasma was separated from whole blood donations in order to produce cryoprecipitate in blood banks. This component allowed widespread treatment of hemoglobin A and increased greatly the life expectancy and quality of life of patients with hemophilia A. The remaining plasma was sent to public fractionation agencies for the production of plasma-derived medicinal products. This situation has continued, even though the production of cryoprecipitate has slowed down considerably, and most fractionation companies are now private enterprises. I now want to make some remarks about blood systems globally. In this paper from The Lancet a few years ago, it was concluded, following an analysis of blood supply and demand, that many countries in Africa, Asia, and Oceania have insufficient blood to meet their needs. I need scarcely add that the first and highest priority of a blood service is to collect sufficient blood for everyday hospital-based medical and surgical interventions. This graphic from the same paper reinforces this fact, showing that, like the WHO data which I showed earlier, many of the less economically privileged countries have a high need to supply ratio, indicating inadequate blood volumes collected. And when assessing this issue of transfusion needs, we need to understand that these vary from country to country and that this variation can be very significant when comparing low to high income countries, as we can do with this graph comparing the transfused populations of Benin, a low income country, and Denmark, a high income country. In Benin, the majority of blood is transfused for hemorrhage, in obstetrics and trauma. Large volumes are also transfused for malaria, a form of anemia associated with blood volume loss, as well as red cell depletion. In Denmark, a wealthy health system uses most of its blood collections for mainstream surgery and the treatment of cancer patients. In Benin, many of the transfusion needs are best met with whole blood, while in Denmark, components are more often the treatment of choice. Despite these country-specific features in the composition of the transfused population, we have seen Western influences in the form of aid to blood services in low-income countries, which can be debated. In this paper, authored by many transfusion authorities, it is shown that countries receiving aid were much more likely to transfuse components than whole blood, despite the transfusion needs noted in the previous example. Clearly, the aid-giving agencies, such as the PEPFAR program of the United States in the first years of the millennium, were implanting the production practices of the donor countries into the recipient countries. It is doubtful that this reflected these countries' clinical needs. The provision of some level of whole blood in clinical transfusion practice is not limited to low-income countries. In recent years, the transfusion of whole blood in massively transfused trauma patients has been revived in Western countries, with the realization that patient resuscitation with whole blood decreased mortality compared to resuscitation with individual components. This is shown in this slide from a recent paper in the Annals of Surgery. The huge volumes of plasma produced from the separation of all blood donations into components find limited clinical utility in transfusion practice. This systemic review from the Cochrane collaboration is just one example where the evidence for the use of fresh frozen plasma, in this instance for cardiovascular surgery, is not evident compared to controls. So now we come to the proposition that the first essential stage of the development of a blood service is the establishment of an infrastructure for the collection of sufficient volumes of safe blood for transfusion. As medical needs start to draw in more patient populations, red cells for normophilemic anemia, platelets for cancer patients, etc., will require the production of components separated from whole blood. The next stage needs to address the needs for plasma-derived products from plasma produced in excess of hospital needs, and later from plasma collected specifically for this purpose. This plasma is first best fractionated through contractual arrangements with the manufacturer, although a domestic fractionation program may ultimately also be considered. Such a program for a fractionation plant needs to be realistically aware of the challenges faced by such a project. This slide lists some recent projects which have encountered difficulties. There are vast areas of the world unserved by fractionation plants, and regional cooperation in the establishment of these plants is strongly to be encouraged. And as with the transfused population, the needs for plasma-derived medicines varies greatly between countries, depending on economic status. This is shown in this slide, comparing the needs of Indonesia and France. In general, low and middle-income countries still use a considerable volume of albumin compared to other products. In the high-income countries, the treatments requiring immunoglobulin dominate the plasma therapeutic landscape. Plasma-derived coagulation factors are still widely used in low and middle-income countries, but have been substituted by alternative therapies in high-income countries. Addressing the safety issues around plasma products, we need to start with ensuring the safety of the starting plasma from infectious agent transmission. We need to understand the way in which viruses replicate and infect a donor's blood, and develop strategies to address the different stages of viral infection, including the pre-serological window period, as shown on this slide, before serological tests can detect the antibodies generated against the virus. The choice of testing platform for detecting viremic donations in the serological window period influences greatly the ability to exclude such donations from the blood bank inventory and the plasma pool for fractionation. This is shown on this slide from the relevant WHO guidelines. Nucleic acid testing is the most sensitive platform. Note that certain combo assays, as well as rapid detection tests, are less sensitive and result in longer window periods. It is the case that, once again, economic status influences the choice of tests to close the window period. In this paper from an Indian blood center, use of nucleic acid testing results in a considerable reduction in the risk of infection with the most important transfusion-transmitted viruses. However, most Indian blood centers are lacking access to this methodology, with only 7% of the total blood collected in India being tested by NAT. Another route to increased safety is through processes designed to destroy contaminating pathogens. In this study from Uganda, the effect of combining pathogen reduction of blood units with different types of rapid detection tests is shown. I emphasize that this is a modeling study, as pathogen reduction of whole blood is still not approved. As you can see from this slide, the two measures augment each other. Turning now to plasma-derived medicines, we assess the measures implemented for their safety through the concept of the so-called safety tripod, whereby we view the three measures which contribute safety to donated blood, which are selection of safe donors, screening of blood donations, and pathogen reduction when available, in relation to the provision of plasma products. Because plasma products are manufactured from pools of plasma from thousands of different donors, selection and screening are not enough for ensuring safety. We rely strongly on viral reduction steps, designed specifically for this purpose and incorporated into the manufacturing scheme to ensure safety from transmission. These measures have resulted in the safety of plasma products now even exceeding the safety of blood components, principally because of the ability to perform viral inactivation on all plasma products, whereas processes for blood components are not yet fully implemented. In this slide, infections from reported infection from a range of arboviruses are shown for blood component recipients. None of these agents, I emphasize, have infected the recipients of plasma products in all the years of surveillance for these viruses, no doubt because of the inclusion of viral inactivation in the process of manufacture. I now draw attention to the requirements for the proper assessment of plasma-derived medicines by regulatory oversight, which is very important. In regards to the quality of the plasma raw material, these requirements include the regulatory status of the plasma supplier, the epidemiology of the donor population in relation to the viruses of interest, donor exclusion criteria, the screening tests which are done on the blood and on the plasma, quality assurance measures, inventory hold of the frozen plasma before it is initiated into the process of manufacture, the size of the plasma pool, as well as the testing of the plasma pool. This slide lists the important aspects in relation to the manufacturing process. These include critical manufacturing steps and related in-process controls, the viral inactivation and removal steps, which I have emphasized before, testing for process consistency, as well as the batch release specification according to a predetermined assessment of quality and predetermined indices of such quality. And finally, the regulator must also have information on the product itself, including its potency and shelf life, all the markets where the product is available, the history of the product since its initial market approval, as well as importantly clinical studies demonstrating the product's efficacy. I will now address the issues around some of the principal plasma-derived medicines, starting with albumin, which, as we have seen, is frequently the major product used in low and middle-income countries. Albumin was developed during the Second World War as a resuscitative fluid to treat blood loss and hypovolemia from battlefield injury. In the succeeding decades, new techniques in traumatology and intensive care have led to the development of alternative protocols using crystalloid solutions, which, when compared to albumin, result in equivalent outcomes, as shown by this meta-analysis from the Cochrane collaboration. However, other indications for albumin have emerged, using the pharmacological aspects of the albumin molecule. These have included the treatment of cirrhosis, which has been studied extensively in clinical trials. These have yielded different results, as shown from the two studies published in The Lancet and the New England Journal of Medicine, which I show on these slides. The Lancet trial suggested a survival benefit for cirrhotic patients given albumin, which was not confirmed in the New England Journal's trial. We are still uncertain, therefore, as to albumin's exact benefit in cirrhosis and hepatology. Turning to coagulation factor VIII, we see the benefit of access to concentrates of this product in prolonging the life of patients with hemophilia A, as reflected in the prevalence, which increases with availability of product. I mentioned earlier, plasma-derived factor VIII has mostly disappeared from the high-income countries, where it has been replaced by synthetic molecules. Significant amounts, however, are still used in low and middle-income countries, and that there is still a role for plasma-derived factor VIII is shown in this slide, showing the results of a randomized clinical trial comparing the incidence of inhibitors to factor VIII in previously untreated patients given plasma-derived or recombinant factor VIII. The incidence of inhibitors is higher with the recombinant product. This is an important result, as following the elimination of viral transmission in today's generation of products, the development of inhibitors is the most significant adverse effect of factor VIII substitution therapy. I want to make you aware of this book, available freely from the website of the World Federation of Hemophilia, which examines in detail the issues around the safety, quality, and efficacy, which are important for making a choice for a factor VIII concentrate to treat patients. This book was developed to assist countries with limited regulatory resources in coming to a choice as to which concentrate to choose in today's market of products for hemophilia A and B. I now come to immunoglobulin, and this product is the most important in the high-income countries. The availability of this product has had a very marked enhancement in the survival of people born with primary immune deficiencies. This slide shows this for Bruton's X-linked agammaglobulinemia and for common variable immune deficiency. The demand for immunoglobulin has increased as additional indications have been approved for this product. These have included a number of autoimmune disorders, including some neurological diseases. This meta-analysis from the Cochrane collaboration again shows the benefit of immunoglobulin in the treatment of chronic demyelinating polyradiculoneuropathy, CIDP as it is more commonly known, a debilitating condition which today is one of the highest usage indicators of immunoglobulin. The slide shows the benefit which immunoglobulin imparts on patients with this disorder. As with Factor VIII, I draw attention to this book, available from the website of the International Patient Organization for Patients with Immune Deficiencies, APPLN. This assesses all the issues around immunoglobulins which need to be considered when choosing a product. The last product I want to discuss is the immunoglobulin made from donors, which have plasma rich in antibodies to the rhesus D antigen. This product is the most important plasma-derived medicine because it ensures the health of rhesus-negative women and their babies, as shown in the mechanism illustrated in the left-hand part of this slide. The graph shows the decrease in the condition brought on by D antigen immunization in D-negative mothers, hemolytic disease of the newborn. This data for Victoria, Australia are similar to data in all other populations. I cannot overemphasize the importance of managing to access this product and therefore of accessing plasma which is rich in these immunoglobulins for manufacture. So I come now to some tentative conclusions. Once blood services have satisfied the needs of mainstream transfusion medicine, their remit may be widened to include the provision of plasma-derived medicines. This requires a reliable stream of high quality and safe plasma, which is best directed, at least on an initial basis, to a contract manufacturer. A needs assessment is a crucial component of this process. Pathogen safety, assured through donor screening and viral inactivation, is also a crucial requirement, particularly for plasma products made from large manufacturing pools, but also in relation to blood components. The use of plasma-derived medicinal products should be based on clear evidence of safety and efficacy. This is best ensured through the relevant clinical trials, some of which I have showed you. This assurance requires independent regulatory provision in the countries where the plasma-derived medicinal products are manufactured and used. This brings me to the end of my talk, and I hope that you have found it of some use. These slides are all derived from public information and are available freely through the organizers, should any of you listening to this talk be interested in them. The talk itself will also be made freely available in the public domain. I thank you once more for your attention.